It's a tremendous honor to be here at Technion and a tremendous honor to share the Harvey Prize this year. I'm going to speak today more about the secrets of the human genome. So yesterday I talked about, that should actually say part 2 medicine. Yesterday I talked about biology, and I'm only going to briefly mention just by way of summary what I said yesterday, and primarily focus on medicine today. By way of reminder, there was this Human Genome Project. It's old news by now, but it was good fun, and it's worth mentioning that it was the first time biology really tried to undertake an organized, goal-directed project to build intellectual and technological infrastructure so that everybody could use it. It was first thought about in the mid-1980s. It was argued about for many years, 3 or 4 years, so that we could really figure out was it a good idea and how to really do it. And eventually a clear set of goals were established of creating genetic maps to trace inheritance, physical maps to be able to have all the overlapping pieces of DNA, the whole sequence, to annotate it with where the features of genes were, and very importantly, to make all that information freely and immediately available to everybody without any restriction. So the Human Genome Project was finished multiple times. It was finished in June of 2000 when it was announced, and then it was finished in February 15th, 2001 when a draft sequence was published. Then, of course, the draft sequence was only 90% of the genome, and so the international consortium went on to produce a finished sequence of the genome, and that was announced April 25th, 2003, just about 10 years ago today, almost. That was chosen because it was actually the 50th anniversary of the Watson-Crick paper that came out on that day. And that was published then around 2004. So we have many opportunities to celebrate 10th anniversaries, and we're having a good time celebrating 10th anniversaries now of the human genome. But as I said yesterday, and it just bears repeating, that this whole idea of working together to create knowledge that could be used by everybody didn't just end with the Genome Project. Once we had a base sequence, a scaffold of the human genome sequence, many, many more maps could be created. And what we've seen over this past decade is an amazing explosion of projects to layer information on top of these maps, like many layers on top of a Google map, you know, the traffic patterns and where the pizzerias are and things like that. And we've seen now maps of locations of genes, evolutionary conservation, of chromatin state, of 3D folding, inherited variation, disease associations, evolutionary selection, et cetera, et cetera, et cetera. And every day, more and more information is being layered on top of that map to create many more maps and cross-reference them with each other. And then, of course, with a complete sequence of the genome, we have complete catalogs of all the genes and all the proteins. Well, all always means almost all, but anyway. And it means, therefore, that little signatures of a nucleic acid sequence are sufficient to identify a transcript, and small amino acid sequences of several, 10 amino acids are sufficient to uniquely identify proteins. And so that allows us to do biology much more rapidly by using these short signatures to do mass spec detection or gene detection. In addition, we have seen, and I think really stunningly, because we never expected this, that the business of sequencing has become so much more efficient. It has become about 1 million times cheaper to sequence DNA over the last, 10, 11, 12 years. Really stunning. Few things drop by cost by 1 million-fold, and that has made it possible to collect vast amounts of information. Now, this is all really primarily kicked in the last several years, and we're only beginning to see the real impact of this, but it's really stunning. It means that whenever you can use sequencing to do anything, you should use sequencing. Because if you want to detect any molecular interaction or characterize a species or a population or something, just sequence. It's cheaper to do that often than to do, carefully designed experiments at the beginning. Collect that information, look at the information, and then do the carefully designed experiments. And of course, as the costs continue to fall, they will fall under $1,000. I think that's clear. they begin to have broad roles across medicine, and they may fall to $100 or so. That's a little more speculative, but it wouldn't be shocking in the next 5 or 6 years to see technologies taking it down to $100. And then you're talking about costs that are sufficiently low that, of course, you're going to collect that information on a patient, even if you don't know what you're going to do with it. If there's a chance it might be useful, you would collect that information. That's going to begin to change the way we think about medicine. So yesterday, I talked biology. Very briefly, what I talked about yesterday was the way we were trying to squeeze out the information about what were the components of the genome, the genes in there and regulatory elements and things like that. I talked about comparative genomics amongst 4 species and 50 species, and how by comparing all of these species, we found secrets in the human genome. Very briefly, the subject of yesterday's talk was to look at the things we thought we knew about protein-coding genes, about regulatory sequence, about transposons, about non-coding RNAs, and to comment in yesterday's talk about how all of what we thought we knew turned out to be wrong. That was really exciting and fun to realize that, in my case, I had been teaching MIT undergraduates for more than a decade, a whole series of facts which were now completely incorrect. It's good progress. These all turned out to be wrong. There were, in fact, not 35,000 to 120,000 genes in the genome, but only 21,000 protein-coding genes in the genome. And we could figure that out by evolutionary conservation. It turned out our whole picture of the balance between protein-coding information and regulatory information was completely backward. It turns out that there's not much more protein-coding information and a little bit of regulatory information. But in fact, there's much more regulatory information to run a genome and much less protein coding information. It's almost a 5-to-1 ratio, meaning that the regulation of things is very complex. And much of this regulation is what changes between mammalian species. Evolution is really, over the last at least 180 million years, has been working primarily on this regulatory stuff rather than inventing lots of fundamentally new proteins. We learned that the genome, which was littered with parasites, 50% of your genome is transposons that we viewed as junk and parasites. Well, now we realize that, yeah, well, they may be junk, but they're not entirely parasites because they do useful things. That we've learned that they spread evolutionary innovations across the genome, and that we can see that a lot of these regulatory innovations that have been scattered across the genome have been moved around by transposons, and we should view them more as symbionts. And we learned that a few things we thought were weird, like some RNAs that never made a protein. And we had a couple, there's obviously ribosomal RNA and transfer RNA, but there were a few other such things. Turns out that it's a rule, that in fact, there are not a handful, there's more than 7,000 of these non-coding RNAs that are clearly functional. And they encode RNAs, and I spent most of the seminar yesterday talking about the properties of these RNAs and what their functions might be. So all that was yesterday. What I want to do today is ask, how do we take this information about the human genome and apply it to disease? That's really the subject for today. So it's a very big genome. How do you find what matters for disease? So it's 3 billion bases. Typical gene might have 3,000 bases of information in its transcript. Often a little smaller than that, but about 3,000, we'll make it round here. And a mutation might be 1 base. So the challenge in the 1980s was, how were you going to, even in theory, find the mutations responsible for disease? And a variety of methods were proposed, conceived in the 1980s, which were utterly impractical in the 1980s, but theoretically possible. A set of methods for dealing with rare monogenic Mendelian disorders using linkage analysis. A set of methods for looking at polygenic disorders by using population analyses. And a set of methods for analyzing cancers by using tumor normal comparisons. And all of these ideas were inherent in conversations and some papers and theoretical papers in the mid-1980s, but it has taken a very long time to get to the point where we actually had the sequence and the tools to be able to bring these ideas to life. And that's what I'll talk about today, is where we are on this agenda. So the systematic discovery of disease genes, this is really David Botstein around 1980, publishing the idea that the simple linkage analysis that had been developed for fruit flies in 1913 could be used in humans if only you could use enough DNA polymorphisms to trace inheritance in families. That was the idea, and we'll discuss where we are with that concept. But of course, that wouldn't work if you couldn't really trace Mendelian inheritance. If you couldn't do that, you'd have to resort to other things, to using, in effect, the whole population as if it was families. And here were a set of ideas that several of us, David Botstein, myself, and some others were involved in, thinking about how you could use a whole population as if it was a family. Starting with ideas like using isolated populations like Finland to trace inheritance across an entire population, then growing to how you might use Europe and how you might use the entire world. And then, of course, tumor normal comparison. It was already clear in the mid-1980s with the discovery of several oncogenes that if only we could look at the whole cancer genome and compare where these somatic mutations had occurred and see where they pile up across the genome, we might be able to learn a tremendous amount about cancers. And as I'll come back to, Renato Dubecco wrote a very famous paper in 1986 arguing that we needed a sequence of the human genome in order to study cancer. So all right, so how are we doing from these theoretical ideas that were proposed in the 1980s? Well, let's start with understanding the basis of simple Mendelian traits. So pretty well. 1990, before the Human Genome Project started, there were about 70 Mendelian diseases for which genes had been definitively identified. Most of them had either been most of them had actually been candidate genes that were known from the biochemistry, and then people had found mutations. In a handful of cases, it was the case that people had really done linkage analysis because it was painful. Cystic fibrosis took 5 years of work to identify. But as the tools of the Human Genome Project came along, it was the case that by 2001, there were more than 1,300 Mendelian diseases identified. By today, something like 3,500 Mendelian diseases have been identified using these tools. Now, we're by no means done. There's still about 1,800 Mendelian diseases to go, and just in conversations this morning here at the hospital, it's good to know that physicians here are continuing to identify more Mendelian diseases as we go along, looking at endogamous populations here in Israel, multiple new Mendelian diseases being identified, I was hearing this morning, in Druzim and many other populations. And so it's the case that list will grow, but it's getting easier and easier to catch up. And we can see a day when we will have the vast majority of, well, actually we have the vast majority, we'll have essentially all the Mendelian diseases well cataloged. But what about common diseases? You know, most of those Mendelian diseases are weird, rare things whose names you've never heard of unless you work on them. But the things that affect the general population, type 2 diabetes and Alzheimer's and predispositions to common cancers and such, how are we doing with those? I want to take some time and talk about where we are and where we still have to go with regard to common inherited disease. So, and this I'm going to describe work from many, many people in the Broad community who have really led these efforts. David Altshuler, Mark Daly, Seik Kathreisen, Romnick Xavier, Steve McCarroll, Joel Hirschhorn, and others. So here, in 1990, when the Human Genome Project was just starting, the number of genes that we knew as involved in common genetic diseases was approximately 1. I say approximately because it was the HLA locus, and there are many different associations to different diseases at the HLA locus, but it was mostly the HLA locus, and it was candidate gene looking at autoimmune-related diseases. By the time the Human Genome Draft sequence was done, it had gotten up to a measly 25, not a very impressive increase. Well, there were no systematic tools. All of these 25 were candidate genes that arose from biological insights rather than any systematic looking across the genome. But in the mid-1990s, some ideas began being developed that roughly went like this. You know, it's not such a big population. The human population traces back to a founding population with an effective size of 10,000. 10,000 is not so big. It can't support that much common genetic variation. You can even write down a formula that tells you about how much variation there can be in the population. Why not just collect all the common genetic variation in the human population, where common means, oh, I don't know, 1% or 5%, pick your threshold. And you know, maybe that's 12 million polymorphisms across the genome, or if your threshold's a little lower, 20 million. But you know, collect them all. And then just look at them across thousands of people and see which ones are enriched in which diseases. Actually, a very simple notion. There's a bunch of theory under it, but the idea is very simple. Now, of course, it was also somewhat insane, because at the time you only knew a few thousand polymorphisms, not millions. And you know, if you had to collect millions, and then you had to genotype millions of polymorphisms in thousands of people, which is billions of genotypes, which at the time was done one at a time, and the graduate students objected to the notion of doing billions of genotypes, not unreasonably, in my opinion. So anyway, it was still a good idea, and the question was, how do you get going down this path? And so you put one foot in front of the other, and we found that it wasn't so hard to go from thousands of polymorphisms to hundreds of thousands of polymorphisms to millions of polymorphisms. By the time the Human Genome Project paper came out, it had a companion paper in the same issue that reported about 1.4 million polymorphisms in the human genome. And today we've got something like 20 million polymorphisms identified in the human genome. That turns out that you don't actually have to study each and every one of them separately, because within a region, they're locally correlated. These guys here are sufficiently well correlated that if you know two of them, you know the rest. So if only you knew that local correlation structure across the genome. This picture comes from work of Mark Daly in 2001, who had found this beautiful correlation structure in a particular region of chromosome 5, but it was so clear from the beauty of the picture that it had to be general, that if we knew the structure across the whole genome, we'd be able to get away with looking at only a subset of the polymorphisms. And so the notion was, construct a map of these correlated regions, these haplotypes across the entire human genome, and then we could use it. And so, you know, several of us made a proposal to do this, and an international haplotype map project was launched in 2002, and happily, the first stage was completed by 2006, and we had the correlation structure of genetic polymorphism across the genome, and could use that then to pick out subsets of these variants. So now, instead of having to do 12 million genetic variants, maybe you only have to do 300,000 genetic variants. The graduate students still objected to the notion of doing 300,000 genetic variants in thousands of people, but it was getting better. At the same time, technologies were being developed by several groups, and these technologies let us go from doing 1 marker at a time to 10 markers at a time to 1,000 markers at a time to 1 million markers at a time, and today 2.5 million markers at a time. So then suddenly you could do 1 million markers. They could proxy for essentially all the common genetic variation in the population. And now the graduate students didn't object so much, because you could do 50 in a day, and it wouldn't be that hard to get through thousands of people. So this is what began to happen. We began to go from a world where the number of discoveries of common genetic variants associated with common diseases across the entire world went from an average of about 1 per year worldwide to, in 2005, 4 discoveries. Then in 2006, 8 discoveries. Then in 2007, a lot of discoveries. That was approximately when all the tools came into place, and you could really begin to extract the power. And to today, when many more than 2,000 loci have been identified across the human genome, involved in more than 200 different diseases and traits. So we have a profusion of loci, common genetic variants, that are associated with various common diseases. An early example was age-related macular degeneration, a disease about which in 2004 we as a world knew nothing. Within 2 years, 3 loci had been identified, which together explained 50% of the variance for the wet form of age-related macular degeneration. And by the way, they encoded complement factor H, complement factor B, and complement component 2, making you guess, even if you knew nothing about the biology, that this had something to do with the complement system, in fact, the alternative complement system. And indeed, that is the case. So that's sort of the poster child where the genes immediately suggest the biological process. So that's a beautiful example. Now, many diseases are much more complex and have a lot more genes involved in them. Let me give you an example from work about lipid levels. So Mendelian looking had identified Mendelian dyslipidemic syndromes and a bunch of individual genes in which you could get simple Mendelian forms of dyslipidemia. Now, what about looking more broadly at common genetic variants that are associated with lipid levels but don't cause clear Mendelian syndromes? Well, Catherison undertook a beautiful study in which he looked at a large number of people, and he found some additional loci. In particular, all of those additional loci. More than 100 genes across the genome associated with lipid levels, HDL levels, and LDL levels, and triglyceride levels. Now, these are particularly interesting. I'll pick a couple of examples from just to illustrate. One of them is the sortilin-1 locus. I won't describe its biology, but there are alleles in the population that lower your levels of LDL by about 16% and lower your risk of coronary disease by about 40%. And when you make the mouse knockdowns and overexpressions, they mimic the human phenotype, and you can then begin to study that biology. There's beautiful work from Helen Hobbs, who has studied the PCSK9 gene. In this case, there was some Mendelian examples of families that had gain of function alleles that caused the hypercholesterolemia. And she found common genetic variants, frequencies about 2% in the African American population that were stop codons segregating, that in fact lower your LDL levels. And they lower them quite dramatically by about 21 mg per deciliter and about 50% lower risk of coronary heart disease. In fact, Helen demonstrated that this 2% allele in the African American population was associated with low risk of heart disease, and it suggested this could be an interesting, because this is a circulating protein that regulates the degradation of the LDL receptor, it might be an attractive target for an antibody therapy to be able to deplete this protein and thereby decrease the degradation of the LDL receptor. Is it safe? Well, happily, she found one individual who was a homozygote for this stop codon and completely lacked PCSK9, which then allows you to investigate the safety of not having PCSK9. And this person was an aerobics instructor, suggesting that it was not associated with any severe phenotypes. And so on the strength of that, several companies have developed antibodies against PCSK9, and they have, just with one dose, dramatic LDL lowering effects. Now, there's another interesting thing that you can learn from all of these loci together. And this was a beautiful paper from Catherison and some others at the Broad last year. They were looking at HDL levels. As you know, LDL is bad and HDL is good. That's slightly oversimplification, but what it means is that from an epidemiological point of view, if your LDL level is high, it's associated with risk of heart disease, and if your HDL level is high, it's associated with protection. From this, you might infer that lowering your LDL level is a good thing. From this, you might infer that raising your HDL level is a good thing. You'd, of course, be wrong, because the fact that an association is seen in the population doesn't mean that an intervention is going to make a difference. It's actually an experimental point you have to test, because both of these could be the result of those being results of some other cause of heart disease. So knowing that actually changing your LDL level would have an effect on heart disease wasn't known until the mid-1990s, when the statin studies were done and showed that they actually provided protection. What about raising HDL? Well, pharmaceutical companies said, let's go for it. Raising HDL probably will be the same thing. And 4 pharmaceutical companies have spent more than $1 billion each to raise HDL levels. So, do we have any evidence that raising HDL levels in the absence of an effect on LDL would be a good thing? Well, how could we tell in the absence of a drug whether that would be a good thing? Can we do the study without having a drug? It turns out we can. Because we have all these genetic loci that vary your LDL levels and vary your HDL levels. And people have them from birth. So they have them from birth. We could look at people, that is to say, SAKE could look at people who had inherited HDL-raising alleles which had no effect on LDL and ask whether they had any protection from heart disease. Answer, they have no protection. They confer no protection at all. If you inherit LDL-lowering alleles, that protects you from heart disease. But inheriting HDL-raising alleles has no effect on heart disease. So that's very interesting. It suggests that that is not a useful thing to do on its own. And perhaps not coincidentally, Pfizer's drug that raised HDL with no effect on LDL had no beneficial effect on heart disease. It was actually a slightly dirty drug. It actually had negative effects on heart disease. In fact, now Merck has a drug right now that both raises HDL and lowers LDL, and I suspect it will have a good effect on heart disease because of its LDL lowering component, not because of its HDL raising component, but we shall see. Many, many more examples like this. Work of Mark Daly and Ramnik Xavier on Crohn's disease has identified more than 140 loci falling into very sensible pathways, autophagy and innate immunity and IL-23 signaling pathways. In these cases, various of the alleles have been modeled in mice and reproduce the phenotypes that you would expect from the association with Crohn's disease. So look, this is hard stuff. 140 loci, working up the biology of 140 loci is hard stuff. And most of them haven't been done, but it's really striking that when people really work on these loci, they're finding that they can work through the biology. And another thing to say is these things end up being extremely reproducible. I probably should have said that nobody's interested in a locus whose significance level in an association analysis isn't at least 10 to the minus 8th, usually 10 to the minus 12th. Some of these are 10 to the minus 50th. Loci that have effects like that reproduce. All right. Now, here's a case that was very depressing, schizophrenia. This is a summary of a study of 7,000 samples done by my colleagues at the Broad, Ed Skolnick in particular, working on schizophrenia. It produced 0 significant loci using this hard-nosed standard of significance. That's depressing if you've just done 7,000 samples. Well, on the strength of this, the National Institutes of Mental Health said, let's not fund any more of this stuff. But, you know, anyway, Ed had momentum and kept going. He did 20,000 samples with his colleagues. Well, now there were 6 significant loci. Not a lot, but, you know, 6. But when they began to analyze this, they said, boy, there's a whole lot of loci here that are, you know, maybe trying to be significant. Maybe if we had more samples, you'd see even more. So what's very interesting is this chart. Here's a chart showing the number of loci that have been identified for height and for Crohn's disease as the sample size increased. In this case, the sample size is 5,000 and 5,000 times this factor. Here, it's 1,000 and 1,000 times this factor. But you'll notice the rows kind of match up. So while the basal case differs 5,000 in one case, 1,000 in another case, as you increase the sample size, the number of discoveries you're making is kind of following the same track. So then you look at schizophrenia and you say, golly, it sure looks like it's doing the same thing. What would happen if we increased the sample size by a factor of 3 or by 6-fold? And here, someone at the Broad, Stefan Ripke, was giving a talk on this, and I made him promise me how many loci he would discover if he increased the sample size. And doing the calculation, he said that he should see 60 loci. So he actually tripled the sample size and found 62 loci. That's actually scarily close. There's some sort of general curve that's going on here, which is very interesting. And the sample size has now increased. They've now looked at 30,000 cases and 50,000 controls. And the current scorecard is they've identified 93 loci in schizophrenia. Most of this is unpublished. About 60 of them, I think, have been reported. And they're beginning to be very interesting. 4 loci affect 4 subunits of the same L-type calcium channel. I think that says something. A bunch of the loci affect a particular microRNA, miR-137, and a bunch of target genes for miR-137. And this microRNA modulates proliferation and differentiation of adult neural stem cells. And again, I think that's saying something. So it's kind of interesting to see all of this stuff emerging here. Now, sometimes you get cool surprises, like a study of diabetes and heart disease hit exactly the same region. And you might say, oh, diabetes, heart disease must have something to do with lipids. Region has nothing to do with lipids whatsoever. But then more diseases got done, and lots of things piled up in exactly this region. And you began to see hits to all sorts of cancers as well. And what's in the region is the cell cycle genes CDKN2A, 2B. And all of these hits are to the same region, but to different little pieces of the same region. And what's clearly being hit are different regulatory sequences clearly being hit. I believe that these are different regulatory sequences across the region, and you're looking at the different enhancers affecting these cell cycle genes, and various people are working that out. So we're not close to done. So far, all that's been done is looking at the high-frequency common alleles, things above 5% in the population. There's a lot more to go. Now, there's a lot of hand-wringing and worrying. There's hand-wringing that says, but oh my goodness, most of the discoveries involve loci that increase things by only 10 or 20%, your risk by 10 or 20%, and that's not a lot. Well, that's actually roughly what epidemiological factors do very frequently. But people say, well, you know, it can't matter. If things don't have a big effect, why are they important? Well, the effect of the allele that's segregating in the human population tells us nothing about what the effect might be from a pharmacological intervention. Good example. HMG-CoA reductase is the site of one of these loci associated with LDL, and a common variant in the human population affects your LDL levels by a measly 3 mg per deciliter. You would say "feh," right, to such an effect. But of course, HMG-CoA reductase is the target of the statins taken by about 35 million Americans. It's clearly a very important locus, and the fact that the common variant in the population has a small effect has no bearing on the fact that it's a superb drug target. So one can't make that connection there. The other thing that we worry a lot about, and people worry a lot about, is when you add up the amount of heritability that is explained by all of these loci, it still doesn't explain most of the heritability of the disease. So where's the rest of the heritability of the disease running around? Now, it turns out as more and more samples have been done, the heritability is increasing. It's going up into the 20% or so. But you know, where's the rest of the heritability? Well, I think there are several answers to the question. The first answer is, it's clear by indirect inference that there's a lot more heritability to be explained by common genetic variants. And you can tell it by looking at the ones that are sub-significant. The ones that are sub-significant, you can make inferences as to how much they're trying to explain. And it looks like for many of these, it's going to get up into the 30, 40, 50% range explained there. But another thing to be said is we haven't begun to look at the rare genetic variants. And there's a tremendous amount of hope about the rare genetic variants. Now, there's not been that much work done. There's only about, I don't know, 7, 8 loci, where there's 7, 8 diseases where there's been any work on rare genetic variants in any kind of systematic way. There are no genome-wide discoveries that have been reported yet. There have all been sets of genes identified for other reasons. But there's hope that they will explain a significant fraction as well. But maybe they're not going to explain everything. Maybe with all the common and the rares, it still doesn't look like it might add up to 100%. Now, how can that be? Because if you have all the commons and all the rares, you're done. There's nothing more to be had. Well, I'm going to digress. You'll forgive me to digress for a second if this seems You can ignore this little digression if you want to, but I'm going to do it anyway. What do we mean by explaining heritability? We often say, we've only explained this much of the heritability. What is heritability? Heritability is a calculation. It's a little calculation you do. And it involves two things. It involves a kind of bottom-up calculation. I'm going to spare you the details of the math, but this is fundamentally a mathematical point. When I discover 100 genes for something, for each one I calculate what its effect is. It has a certain effect, beta. From that, it explains a certain amount of the variance. You could do a one-dimensional variance calculation and see how much of the variance is explained by that variable. You add them all up, and you get the variance that you've explained so far. You then compare it to the total variance that you should be explaining due to the additive effects of loci, and you calculate that by the correlation between relatives in the population. So there's one number, a bottom-up number, which is called h squared all, of all the effects of the individual loci, and there's a top number, h squared population, and you calculate that, and you see how close you've gotten. And there's a beautiful theorem, and the theorem says, if the trait involves no genetic interactions, they equal each other. And that's why you compare the two. The problem is it says if the trait involves no genetic interactions. If the trait involves genetic interactions, the theorem is false, and the two numbers shouldn't equal each other. This is actually a serious problem, because if it's false, we have no idea how much it should add up to. And so all the calculations we're doing, where we're wringing our hands that we haven't explained everything, we mean we haven't explained everything that we should be explaining, assuming there are no genetic interactions. So there could be a lot of this due to genetic interactions, and that's an important part to keep in mind too. So for all of those reasons, what we really, and this is not a small point, I'll say for Crohn's disease, for the 74 loci we analyzed for Crohn's disease, if there are no genetic interactions, they explain 22% of the variance. If there are genetic interactions that work through 3 pathways coming together, you can write a simple model where those loci explain 58% of the variance. So what that should do is destabilize you with regard to any of these variance calculations. The truth is, I don't really believe the variance calculations. I think one has to distinguish between why you're doing this. If you're doing variance calculations to make personalized risk prediction for each patient, well, then that matters how much you've explained. If you're doing if what you're doing is doing these studies to understand the biological pathways, you don't care how much variance you've explained. It's the loci you've identified. So there's a very different set of goals, and whether you're trying to do personalized risk prediction or biological pathway identification. And I think that's an important distinction. Anyway, that's what I'll say about inherited disease. Now let me turn for the next bit of this talk, if I may, to cancer. So cancer is another pretty complicated situation where the hope was, so I think we're making good progress on the inherited disease stuff. We're going to need larger and larger samples, et cetera. But what about cancer? Understanding the cancer genome. Well, the idea that cancer was a disease of the genome was an old idea. It goes back to 1914, to Boveri, who said that chromosomal problems might underlie cancer. Of course, that was forgotten for a long period of time through the 20th century, when everybody was focused on the idea that viruses would probably cause cancer, so much so that by 1970, in many reviews, but I picked one particular statement from a review in 1970. Someone wrote, "The viral origin of the majority of malignant tumors is now documented beyond any reasonable doubt." And you should always worry when people start invoking beyond a reasonable doubt, legal standards or things like that in scientific papers, because within a few years it became clear that this was completely false, and that in fact the pendulum swung back to understanding that the genome was really the source of the vast majority of issues in cancer, with the discovery by Bishop and Varmus of viral oncogenes related to cellular genes, and by the mid-1980s, the discovery of critical genes, RAS, ABL, MYC, retinoblastoma, mutated in all different ways. Now, by this point, as I've already referred to, Renato Del Becco, I think, really crystallized it by saying this is cool, but there's probably a lot more going on, and trying to discover all these genes retail, one at a time by hand-to-hand combat, is going to be hard. We need a whole sequence of an organism, preferably the human. And so his call in 1986 was one of the very important calls to get a sequence of the human genome. So cancer work went on, the genome went on in parallel, and you get to the early 2000s, and now it was finally time to really see whether this sequence of the genome was going to be very useful. So a bunch of us got involved in a National Cancer Institute working group in 2005, which proposed the idea that we should kind of have a cancer genome project that would systematically take hundreds and then thousands of tumors and sequence and compare them to the matched normals, identify where the mutations were, and begin to enumerate all the genes that could be hit and mutated in each type of cancer. Now, as happens with every major project, it then is subjected to debate in the scientific community. There were a number of objections to the project. The objections were, number one, all of the cancer genes are already known. Objection number two, there are so many cancer genes that we'll never know them, it's hopeless. Now, you would wish that you could get those two people to argue with each other first, so you only had to argue with the survivor, but in any case, you don't. You have to answer both objections. And then there was the canonical, the cost is too high. And that's also a serious objection. You shouldn't be spending too much money on these things. But eventually, a sensible plan was put together, and it was decided to do a pilot phase and then a full phase. You know, it didn't necessarily, you know, there were serious objectors to this. This is one of my favorite objections. The Human Cancer Genome Project, one more misstep in the war on cancer, a favorite of mine there. But anyway, people came together around these pilot projects, a Cancer Genome Atlas in the United States, an International Cancer Genome Consortium amongst 15 countries around the world. And, well, as better and better sequencing tools came along, it was possible to start interrogating the genome. You could, by just doing this large-scale, cheap, massively parallel sequencing, you could look for point mutations, amplifications, deletions, translocations. Roughly speaking, where are we now? Well, at the Broad, we've done, actually, now it's probably more than 4,000 tumor normal pairs across a wide variety of cancer types. And what we see is tremendous heterogeneity in many things. It's really interesting. When we wrote a proposal, a bunch of us, for a cancer genome project, we said the average mutation rate in cancer is 1 in a million. Well, in fact, it's wildly different. 1 in a million sort of in the middle there. Some tumors are much lower. Pediatric tumors are much lower. Mutagen-induced tumors like skin cancer from UV or smoking, much higher. But even within a tumor class, there's huge amounts of variation in the overall rate of mutation. Not only that, but the patterns of mutation are wildly different too. You can, for every tumor, write down the kinds of mutations that you're seeing as a function of the flanking base context. And then you can compare them across tumor types and find that different types of mutations, CpG deamination, more common in certain types of tumor types. The kinds of mutations you associate with the ApoBec process here. Mutations here, C to T mutations in UV-induced cancers. Smoking cancers, very different patterns of mutations. You can see wildly different patterns of mutations with regard to how amplified the genomes are, deleted the genomes are, rearranged the genomes are. You can find all sorts of crazy new processes. There are times that chromosomal catastrophes happen, where a single chromosome is shattered into lots of pieces with all sorts of very few translocation events in most of the genome, but then dozens and dozens of events shattering single chromosomes. Or beautiful daisy chains, where A goes to B, B goes to C, C goes to D, and you get a series of 8 translocations connecting different regions of the genome, indicating that there were simultaneous catastrophic events where many pieces were broken and were stitched together. So you're getting a picture of tremendously different processes giving rise to cancer. But all right, what do you learn about the genes? Well, the earliest studies were, I think, encouraging. The early studies, before you could do whole genomes, Matthew Meyerson at the Dana-Farber and the Broad identified mutations in the EGFR gene by looking at 50 receptor tyrosine kinases and found mutations in the EGFR gene in lung cancer. And it turned out that those were associated precisely with the patients who responded to EGFR inhibitors. But that was, limited to 50 genes, and you needed a good guess. What's been really cool is seeing what happens now when you can take a totally unbiased look across the genome. So I'll give you an example from multiple myeloma. In multiple myeloma, when you look across the genome in multiple myeloma, well, before we knew that KRAS, NRAS, and P53 were mutated in multiple myeloma. When you begin to look systematically across the genome, you find there's a tremendous amount more going on. There are mutations in 40% of the cases affecting genes involved in protein homeostasis, all sorts of genes involved in protein homeostasis. 25% of the cases involve mutations in the NF-kappa B pathway. 10% of the cases involve mutations in IRF4 transcription factor or its downstream targets. 4% of the cases have mutations in BRAF. So you end up with, and 16% of the cases have mutations affecting the coagulation pathway, which is really weird because that's an extracellular pathway. But in the bone marrow, effects on coagulation can then come back through activation of PAR1 and potentially be mitogenic. So all told, there's a whole bunch of pathways, 7 new pathways that emerge that weren't previously known in multiple myeloma. Similar sorts of situations emerge in head and neck cancers, where all sorts of new pathways and new sorts of genes emerge. Indeed, by now, and Levi Garraway and I wrote a review, just came out last month, lots of tumor types have been analyzed, and many, many new genes and processes have been identified through this. The processes include mutations affecting metabolism, mutations in such boring enzymes as isocitrate dehydrogenase that cause it to create a new metabolite, 2-hydroxyglutarate, that appears to then inhibit certain enzymes involved in chromatin. Many mutations affecting epigenomic processes and chromatin regulation. There had not been evidence, it was clear that tumors had screwed up epigenetics, but it was not clear that it played a causal role as opposed to being a result. And now it's very clear that there are many hits across epigenetic and chromatin regulators. RNA splicing, lots and lots of mutations affecting RNA splicing. Lots of mutations affecting lineage specification. So the classic mutations affecting signal transduction pathways are obviously critically important in cancer, but there are lots of other processes that are getting affected. And many of them are in things like splicing and translation and protein homeostasis and such. And we don't actually know how they're causing cancer, what their targets are, but it's clear that they're very significantly associated. Now, there's a long tail of genes still to go. It's clear that as you analyze cancers, there are many, many, many more genes that are being discovered here, and we're not close to done. And given that there are, in the United States, 1.5 million new incident cases of cancer a year, there'd be no excuse for not doing larger samples. There's certainly more than enough people who are getting cancer, and we ought to be able to squeeze out that information. In addition, all of this is coming from looking at protein coding regions. Essentially, no work has been done on the non-coding regions of the genome. This, which appeared just in February this year, is the first report of regulatory mutations in cancer. 2 nucleotides in the promoter of the telomerase reverse transcriptase gene that activates it. So there's a huge amount of dark matter that hasn't even been looked at yet. Now, I'm going to skip over some details to simply say there's a lot of work involved in figuring out how you can look at large numbers of samples. And rather than show you the slides, I'll just tell you, as you begin to increase the sample size, the number of significant genes you see begins to skyrocket. And you shouldn't trust it at all, because the genes that you start seeing are weird genes. They tend to be very big genes, and they tend to be olfactory receptors, which should give you pause. A lot of smell receptors being involved in cancer, smells fishy, right? So it turns out that as you increase your sample sizes, you have to be very careful to have the right background mutation rate for each gene. Because if the genes have heterogeneous mutation rates, and you're modeling them as if they have a homogeneous mutation rate, you get excited about any gene that has too many mutations relative to the average rate. You actually have to have a gene-specific rate. Olfactory receptors have a high mutation rate. They're in the late replicating part of the genome. Long genes have large mutation rates. When you correct that, it all's kosher again. But without it, and in fact, there are some things in the literature now that it's clear that they are not significant genes because they weren't corrected for the right mutation rate. But with all that done, and Gadi Getz, one of the many fantastic Israelis at the Broad Institute, has done brilliant work that's coming out in Nature showing how you can correct that and get those rates just right. You're able then to begin to pile together thousands of samples and begin to look at genes that are significant where you might not have seen them in any one cancer, but across the whole set you can see them. And Gadi has gone on so far to find at least 60 new genes that weren't evident in any single cancer set, but together you can see across many of them are highly significant. So, all right. Hang on just a second here. So, what remains to be done? A lot. A lot. We've barely scratched the surface of what's got to be done. We need to complete this whole mutational atlas I've been talking about in primary tumors, but that's just the start. We've got to really get this into the clinic with respect to looking not just at primary tumors, but tumors through the process of progression, through metastasis, response to drugs, which ones respond and don't respond, tumors that become resistant, look at the heterogeneity within tumors through single-cell analyses. We really need to look at the animal models and ask which ones represent good models for human cancers. I think dog models are particularly interesting because they naturally get cancers that can resemble the human. And then we need to go beyond just simple cataloging of mutations. We need to do the same thing with kind of functional catalogs, taking thousands of cancer cell lines whose genomes have been fully characterized and then characterize their sensitivity to inhibition of each gene in the genome and to wide varieties of drugs to make kind of a functional encyclopedia. So we can begin to say that mutations in these pathways confer an Achilles heel, a sensitivity to hitting these pathways. And we need a lookup table that says, given genotype, where are the sensitivities? Because in the long run, this is how we're going to put together as a community the combination therapies that matter, and it's got to be done in a systematic way. And with the ability now to make more and more cell lines using the ROCK inhibitor techniques that allow you to immortalize lines where you couldn't do them before, I think big catalogs can be made. But to make all of that powerful, we need to combine data. And this is a hugely important challenge. That if we try to do this with the data produced at the Dana-Farber, or the data produced at Memorial Sloan Kettering, or the data produced at this hospital alone, we will not have enough power. We need to be able to figure out how to integrate data across the world while protecting patient privacy. I can't emphasize how important this is going to be because we're all going to see rare instances of some great response or partial amounts of data that alone won't be significant and become significant when you combine things. So I think one of the key things, and the last point I want to make, is that we need, in the next year or two, a global alliance. We need a global alliance to be able to combine data. Now, in the end, this has to be with the permission of the patients and the trust of the patients because it's their data. It has to be something where every patient has the right to say, count me in for this project. Have consent forms here at the Technion and at Dana-Farber and Mass General and every place else where there is, consistent with the laws of the jurisdiction, a box that you can say, check that says, count me into this international project. I have cancer now, and I want to help other people. It may not help me, it might help me, but I at least want, as an act of altruism, to make sure that my experience doesn't go to waste. I think it's doable. I think it will require coordination amongst countries. It will require a technical infrastructure to be able to share and store these data in secure ways. And it will require kind of a worldwide movement of patient trust where we say it is a right to be, I mean, it's your right not to contribute. It's your right to be totally private, but my experience is most people would willingly, when they have a serious medical condition like cancer, want to contribute their data to something bigger than themselves. And so many of us at the Broad are very involved right now in trying to pull together both this international coordination, the technical infrastructure, and thinking about how we reach out to patients. And I very much hope, given the unique medical system in Israel and the, in fact, much better medical records here in Israel than in the United States, that Israel will be a part of such a movement. So anyway, that's it. Yesterday I spoke about biology and what we've learned by squeezing information out of the genome. Today I've tried to give you some sense of how far we've gotten with respect to squeezing information about medicine, inherited diseases, and cancer. And I think the answer is we're doing remarkably well, and we're not close to done yet. There's a tremendous amount still to go. And it's very clear that we're going to have to turn the entire medical system into a learning system. And that's not a bad thing. But that's what it's going to take to really bring this thing home in the end. And I think, well, the other thing we've learned is how to study biology. Before this whole genome era, we studied biology alone in our own labs. And that's incredibly powerful. The single investigator working alone at the bench remains an incredibly powerful paradigm. But what we've learned through the Genome Project and through many of its successor projects is the awesome power of people working together in a collaborative fashion, that everybody can help each other, and we can get things done much larger than any of us could possibly do alone. I want to particularly acknowledge some of my great colleagues at the Broad who have led work on inherited disease and on cancer. And as with all talks on the genome, I want to acknowledge that so much of this is the product of people around the world sharing their data, and no talk on the genome or anything from it stands alone from any one institution. So with that, I just want to say thank you again so much to Technion for the great honor of sharing the Harvey Prize, and thank you all for your attention.